Good afternoon, everyone, and welcome to another webinar sponsored by the North American Vascular Biology Organization. I am Enar Cuervo from the University of Illinois at Chicago and a member of the NAVBO Education Committee. I will be moderating today's session. We are pleased to welcome today our speaker, Daniel Greif from Yale University School of Medicine, who will present his work entitled Pericyte ALK5 TIMP3 Axis Contributes to Endothelial Morphogenesis in the Developing Brain. At this time, I'd also like to welcome Julie Davitt, also from Yale University. She will monitor today's questions. The way we handle questions is in two ways. Throughout the presentation, you can type your question into the question box that you can see in your control panel. These questions will be answered at the end of the presentation. So Dr. Davitt will compile and then post the questions to Dr. Greif. At the end of this question and answer period, provided that there's time, attendees will also be able to ask additional questions. So if then you still have a question, please raise your hand by clicking on the hand icon on the left side of your control panel. Then you will be recognized. I will call your name and your mic will be unmuted, and then you will be able to ask your question live. Before we get started, I wanted to go over some logistical aspects. Throughout this webinar, you are able to switch between the phone audio and the computer audio in case you're having a problem. You can see this information in the audio section of your GoToWebinar control panel. If you experience a few words being skipped in the audio, it may be your Wi-Fi connection. In this case, connecting to the internet through the hardwire should remedy the audio. If you experience technical problems, please click on the Help tab at the top of the control panel and then scroll to the bottom of the Help screen for the technical support phone number. This webinar is being recorded and archived on the NAVBO website for future use. So let me introduce Dr. Greif. Dr. Greif received his MD from the University of California, San Francisco. He trained in internal medicine at the University of Washington and at Brigham and Women's Hospital at Harvard University, and in cardiovascular medicine at Stanford. In 2010, Dan joined the faculty and started his independent lab at Yale University and has risen to the rank of associate professor in the departments of medicine and genetics. Dan received the Springer Junior Investigator Award from NAVBO in 2015 and the Established Investigator Award from the American Heart Association in 2019 and was elected to the American Society of Clinical Investigation in 2018. Dr. Greif's lab focuses on vascular mural cells and myofibroblasts in development and diseases of the blood vessels and of the lungs. Today's presentation will reveal a key role for Pericyte ALK5 in regulating brain endothelial morphogenesis and a substantial therapeutic potential for TIMP3 during neonatal brain hemorrhage. Welcome, Dr. Greif.
Thanks very much, Enar and Bernadette, for giving me this opportunity. I want to thank the North American Vascular Biology Organization for allowing me to discuss the work in our laboratory. So, as was mentioned, I'm going to talk about a TGF-beta signaling pathway in pericytes that regulates TIMP3 and contributes to morphogenesis of endothelial cells in the brain during development. So this work was done at Yale University in New Haven, Connecticut in the United States. So as Enar mentioned, my laboratory focuses on vascular mural cells. So in large vessels like arteries and veins, these are the mural cells are muscle cells. That's what coats the endothelial cells in the large caliber vessels. And in smaller caliber vessels like pericytes, like capillaries, the vascular mural cells are pericytes. In addition, we also look at myofibroblasts. And we look at these different cell types in development, maintenance, as well as in diseases. And here I'm showing the members of my laboratory currently, and these people really deserve all the credit because they're doing all the hard work, and I will take any of the blame. So today we're going to focus on work that was really spearheaded by Julie in the lab and was started by Taya. We also had input from Dr. Weatherby in regard to in situ hybridization, and it was recently published in Developmental Cell, and it's entitled Pericyte ALK5 TIMP3 Axis Contributes to Endothelial Morphogenesis in the Developing Brain. So let me give you a little background about neonatal brain hemorrhage. So the germinal matrix is a region in the embryonic brain that's highly vascularized. It's rich in neuronal and glial precursors, and it's located in front of the lateral ventricles. In terms of perinatal intraventricular hemorrhages, this occurs in around 12,000 premature infants annually in the USA and is much more common in low birth weight infants. It's associated with substantial morbidity, including cerebral palsy, cognitive deficits, and hydrocephalus, and also has a high rate of mortality. And in fact, over half of the patients that have this condition and survive develop one of these morbid or more of these morbid conditions. It largely results from a severe hemorrhage in the germinal matrix. And there really are no treatments for neonatal germinal matrix hemorrhage / intraventricular hemorrhage. The only effective modality is preventative treatment with antenatal steroids, so steroids to the pregnant mom. However, there's some concern that, especially with repeated doses of steroids, these may have deleterious effects on the development of the brain or other organs of the fetus and newborn. So let's talk a little bit about parasites and the blood-brain barrier, and then germinal matrix hemorrhage. So as I alluded to in the beginning, parasites are the vascular mural cells that coat the capillary endothelium. The blood-brain barrier is a tightly regulated interface between the vasculature and the brain parenchyma. And the blood-brain barrier consists of endothelial cells, associated tight junctions, parasites, basement membrane, as well as the end feet of astrocytes. And a reduction in the quantity of brain parasites is associated with defects in the blood-brain barrier. In fact, embryonic mice that lack CNS parasites develop brain microaneurysms and an increased permeability of the blood-brain barrier. And this is largely due to an increased transcytosis through the endothelial cells. So parasites are thought to be critical for protection against neonatal intracranial hemorrhage. And in the human mid-gestation brain, the germinal matrix is prone to hemorrhage and has a relative paucity of parasites in comparison to the cortex or white matter. So this germinal matrix is deficient in the number of parasites that it has compared to other regions of the brain. So we focused on the TGF-beta pathway, and really little is known about how it signals in parasites and its role in parasites, and especially in the context of brain development. So there are, in terms of detecting TGF-beta ligand, the receptors are heterodimeric. They consist of a TGF-beta receptor 2, and then in endothelial cells, they have one of two types of type 1 TGF-beta receptors, either ALK5, shown here, or ALK1. However, parasites only express ALK5. They don't express ALK1. And ALK5 deletion in endothelial cells results in intracranial hemorrhage by embryonic day 11.5 and embryonic lethality. So once TGF-beta is signals through the ALK5 TGF-beta receptor 2 heterodimeric receptor, that will then induce phosphorylation of SMAD2/3, which will lead to recruitment of the common SMAD4 protein, and then this complex will move into the nucleus and modify gene expression. Similarly, ALK1 and TGF-beta receptor 2, when these engage ligand, they'll phosphorylate SMAD1/5, recruit SMAD4, and move into the nucleus. So it's been shown that deletion of this SMAD4 gene in endothelial cells results in perinatal intracranial hemorrhage. And in the human mid-gestation brain, in comparison to the cortex or white matter, the germinal matrix has reduced levels of the TGF-beta 1 ligand. So perhaps the germinal matrix is more prone, at least partially, to hemorrhage because of the reduced levels of TGF-beta ligand, as well as the reduced levels of parasites. And so what we focused on in this study is what is the role of the parasite TGF-beta signaling in regulating parasite biology, crosstalk with endothelial cells, blood-brain barrier development, and hemorrhage in the germinal matrix that extends into the intraventricular compartment. And so what we need to do first is show you a little bit of the anatomy here. So this is a coronal section of a murine brain at embryonic day 12. And you can see the nature of the section here. And marked in red here in L and M are the regions of the ganglionic eminences, the lateral and medial ganglionic eminences. And this comprises the germinal matrix. LV is the lateral ventricles, and this is the third ventricle here. And you can see they're on both sides. And the ventricles, of course, is where the cerebrospinal fluid is. And so again, the ganglionic eminences make up the germinal matrix. And so if we look in our studies, again, doing coronal sections here through this region of the brain between embryonic day 9.5 to embryonic day 12.5, in this region here, you begin to see the germinal matrix getting protrusions here, and these become the lateral and medial ganglionic eminences. And then at embryonic day 12.5, it looks a lot like the schematic did, where we have the lateral and medial ganglionic eminences that make up the germinal matrix, the lateral ventricle, as well as the third ventricle. And when we stain in these regions between embryonic day 9.5 and embryonic 10.5 on sections with isolectin B4, which is a marker of endothelial cells, NG2, which is a marker of mural cells, and DAPI, which marks nuclei, you can see at embryonic day 9.5, there really is not a vasculature at this time point. At embryonic day 10.5, you begin to see isolectin B4 endothelial cells here, but you don't see much in terms of the NG2 positive mural cells. And then this changes by embryonic day 11.5, a dramatic change, and then most of the endothelial cell marking here is covered by NG2 positive pericytes at 11.5 and 12.5. And that's quantified here. You can see there's a marked change between 10.5 and 11.5. So in these studies, we use the PDGF receptor beta Cre. PDGF receptor marks pericytes in the germinal matrix region. And we use that to induce recombination of a Cre reporter, in this case, the MTMG reporter that was generated in Lucien Lowe's lab at Stanford. And so when Cre is expressed in the cells that make PDGF receptor beta, you can see that the cells would be marked with the GFP here. And in our studies, the M-tomato signal is quite weak compared to our staining with antibodies in the red channel, so we don't pick up the M-tomato stain. And so what we did first, because we used PDGF receptor beta Cre widely in these studies, we confirmed that we were getting specific marking with this. And so we used mice that I just explained that had the PDGF receptor beta Cre and one copy of this MTMG reporter, and analyze them at embryonic day 10.5. So remember from the previous slide that at this time you have the IB4 positive endothelial cells, but you really don't have much in terms of mural cells, no NG2 staining in this region here. And as expected, there's no GFP here because we don't have the PDGF receptor beta Cre active now. By the next day, where we had the NG2 coating most of the endothelial cells, here we do have the marking by GFP, suggesting that there's a faithful marking of pericytes at embryonic day 11.5, and that's true at 12.5. And here we have a control where we don't have the Cre, but we have the Cre reporter, the MTMG, and as expected, we don't have GFP expression at this time because we don't have a Cre to induce recombination between the LOX-P sites that allow the GFP to be expressed. So in this region, it looks like our MTMG reporter is not leaky, so that's good. And then we also confirmed that our marking with the MTMG in the presence of the Cre marks PDGF receptor beta positive cells. And in this region, this will be pericytes again, so this will be similar to the marking that we see with NG2. And here the green cells are mostly marking the PDGF receptor beta positive cells. And again, we don't get any marking in the absence of Cre in the GFP channel. So it looks like the Cre is working well in terms of marking pericytes. And so now we use this Cre to delete ALK5, which, as I mentioned, is the type 1 TGF beta receptor that's present in parasites. And so what we did here initially is we took PDGF receptor beta Cre ALK5 flox mutants, and we found that these developed brain hemorrhage. And so parenthetically, actually, what I first noted when I started these studies myself is I was trying to figure out what would happen if you deleted ALK5 in the PDGF receptor beta cells, if that would have an effect on pulmonary artery development. And I found around this time frame that I couldn't detect any change in pulmonary artery development, but it was quite obvious when I looked at the embryos by whole mount that there was a brain hemorrhage. And so later on we followed up on this. Julie really followed up, and Taya followed up on this. And what we found in the mutants here by embryonic day 12.5, you can see that there's a hemorrhage here in the brain, and it gets bigger by 13.5. And so we quantified that by basically taking an area of the head, if you will. So below the snout, we drew a line back, and this area was the total area. So this would be the denominator, and then in the numerator we look at the area of the hemorrhage, and we took this ratio or percentage, and we quantified the mutant hemorrhage area in the percentage, and we saw that it increases on these growth stains between embryonic day 11.5, 12.5, up to 13.5. And then if we look on sections of the control mice, so controls here are always going to be PDGF receptor beta Cre, have the ALK5 flox over plus, and the mutants are always throughout the talk are going to have this PDGF receptor beta Cre and be ALK5 flox over flox. And so we see in the mutants that there's a hemorrhage at embryonic day 12.5 in the germinal matrix, which we don't see in the controls. And then if we look at embryonic day 13.5, a day later, you can see there's a marked upregulation of the hemorrhage in the germinal matrix in the mutants, as well as a hemorrhage in the ventricles. And so it's critical when we look at this that we're not getting deletion of ALK5 in endothelial cells, because as I mentioned in the introduction, deletion of ALK5 in endothelial cells has been shown previously to induce brain hemorrhage and cause brain hemorrhage. And so this was something we spent a long time on to solidify. And so I'll show you some different approaches that we used to clarify this. So this was the first approaches we just did in these PDGF receptor beta Cre mice that had a copy of, in this case, the reporter, the ZS green reporter, one copy of that reporter. And what we did is we took sections and we stained them in the germinal matrix with ERG, which is a marker of endothelial cell nuclei, ZS, which is the Cre reporter, IB4, which is a marker of endothelial cells, and of course DAPI is the nuclear marker. And you can see this endothelial cell here is shown with a positive ERG nuclei, but the ZS is not marking that cell, it's marking the adjacent cell, which is the mural cell in the parasite. We also, from these cells, we isolated endothelial cells with anti-CD31 beads. We looked at the transcript level of endothelial markers, cadherin 5, as well as PCAM1, which encodes CD31. And these were high relative to expression of the transcripts for CSPG4, which is also NG2, PDGF receptor beta in mice, and heavy chain 11, which is a muscle myosin heavy chain protein and markers from muscle cells. And we also isolated these cells and cultured them with FACS. We isolated ZS positive cells as a positive control. And here we isolated endothelial cells from these mice, and we could see that these cells did not express ZS. So these studies suggest that the PDGF receptor beta Cre is not marking endothelial cells, which is critical. It's marking parasites. We went further now, and then we took the parasites, and we found that when we isolated them from mutant PDGF receptor beta Cre ALK5 flox embryonic brains, these have reduced ALK5 levels. But when we isolated endothelial cells from these brains, they didn't have changes in their ALK5 level. So to go through this, these two graphs here are in brain vascular cells, isolated. So we isolated endothelial cells, and we found that the transcript levels of ALK5 were unchanged. And when we isolated parasites from the brain, we found that ALK5 was markedly reduced at the transcript level. When we looked at endothelial cells from other organs, lungs and liver, here again, we found that there was no change in ALK5 levels in the endothelial cells. And then when we looked at ALK1, which again is that other type 1 TGF-beta receptor that's expressed in endothelial cells, but really not expressed in pericytes, there was no reduction in its level in endothelial cells. So in these mice that have the PDGF receptor beta Cre and a floxed ALK5, we're reducing the levels of ALK5 in pericytes, but really having no effect on ALK5 in endothelial cells and having no effect on ALK1 in endothelial cells. So furthermore, we then looked at the signaling in the TGF-beta pathway, and by looking at phospho-SMAD3, and here we found that there was no difference in the percent of endothelial cells in the germinal matrix vessels that are phospho-SMAD3 positive in controls versus mutants. So again, the controls are flox over plus with the Cre, the mutants are flox over flox with the Cre, and here we're looking at endothelial cells. So cadherin 5 marks endothelial cells, as does IB4, and phospho-SMAD3, which we showed in the beginning is downstream of ALK5 in the pathway. What we see is the level of phospho-SMAD3, the percent of endothelial cells that are phospho-SMAD3 positive is not changing between controls and mutants at this time point. So all these things point to that the PDGF receptor beta Cre is marking the pericytes and not the other cell types. So here we also need to consider the marking of smooth muscle cells. So on the left side here, I'm looking at PDGF receptor beta Cre with a copy of the ZS reporter. And you can see within the dashed lines, this is the region of the germinal matrix, and this is outside where my arrow is here of the germinal matrix. And here you can see staining for smooth muscle actin, which is a marker of smooth muscle cells. So within the germinal matrix, we really only have capillaries. We don't have cells that vessels that are big enough to be coated by smooth muscle cells. And so outside here, you can see a larger vessel, presumably an arterial, that's marked by smooth muscle actin, but that does not mark the cells that are within the germinal matrix. And then the other thing to note over here is that the ZS with the PDGF receptor beta Cre is marking the smooth muscle cells. And so now we're using, on the right side here, the smooth muscle actin Cre with the MTMG reporter. And when we combine this, so the mice have these genes as well as being floxed, have 2 floxed alleles for ALK5. These mice do not get brain hemorrhage, as you can appreciate here, as well as below here on section. So this suggests that in order to get a hemorrhage in the germinal matrix, you need to induce recombination of ALK5 in the pericytes, because when you induce recombination in the smooth muscle cells, it does not by itself induce recombination. And we show here that with this smooth muscle actin Cre ER with the MTMG reporter, the GFP is marking the smooth muscle cells, but it's not marking the capillaries that are within the germinal matrix, the small caliber vessels. NG2, which is important to note and is often a error in the literature, is NG2 marks capillary pericytes, but it also marks arterial and artery smooth muscle cells as well. So this will not differentiate those 2 types, at least not in the brain as we show here. So again, this speaks to that what's important in order to get the brain hemorrhage is deleting ALK5 in the pericytes in the germinal matrix. So let's now begin to characterize the phenotype in some more detail. So what we found is that the mutants in their germinal matrix, these vessels have a leaky blood-brain barrier, which is probably not very surprising given that these mutants get hemorrhage. So these mice have the PDGF receptor beta Cre, have a copy of the ZS, and are either flox over plus, which is the control, or flox over flox, which is the mutant. And the way this assay is done is a 10 kilodalton tracer is injected into the liver. This tracer is labeled by rhodamine, and what is known from prior work is that the blood-brain barrier is formed a little bit before embryonic day 15.5. And so what happens when this tracer is given at 11.5, sorry, a little bit before 15.5, I should say. And so what has happened when the tracer is given at 11.5, it just leaks out into the brain as you don't see it intravascular here. But if you give it a little bit before or at 15.5 and the blood-brain barrier is formed, the tracer stays intravascularly and doesn't leak out in the controls. However, in the mutants at embryonic day 15.5, we don't see much intravascular tracer because it has leaked out, suggesting that the blood-brain barrier is not solidified by that time point. We also then begin to characterize the morphology of the vessels in our mutants compared to our controls. So if you look at the top here, at embryonic day 11.5 and staining with cadherin 5, which marks endothelial cells, NG2, which is the mural cell marker, you can see in controls there's quite good coverage of the endothelial cells with pericytes, both at 11.5 as well as at 13.5. However, in our mutants, which have the ALK5 gene deleted, you can see a lot of endothelial cells shown in with the CDH5 green stain here that don't have the NG2 covering. This is shown with the arrowheads here as well as below down here, and that's quantified here. And you can see by 11.5 and 13.5, there's a reduction in the coverage of the endothelial cell vessels by the pericytes. Now, if we look at the vessel parameters at these different ages, these mutant vessels will have a larger vessel area, so that's just the amount of area that's stained by cadherin 5. The area of the lumen of the vessels is larger in the mutants, and the vessel diameter or the thickness of the vessels is larger in the mutants compared to controls. So if we go back up to here and see a reduction in the coverage by pericytes, one possible explanation for this is that the pericytes could be less proliferative. And so we looked at that, and what we found was that pericyte proliferation is unchanged actually in the mutant germinal matrix vessels. And so here we're looking at mice that have the PDGF receptor beta Cre, have a copy of the MTMG reporter, and are either flox over plus or flox over flox for ALK5. And we're detecting pericyte nuclei or cells here basically by using the GFP stain to detect pericytes. These studies were done following a 6-hour chase. We gave BRDU, waited 6 hours, and then sacrificed the animals. And then staining with ERG, which is a specific marker for endothelial cell nuclei. So for pericytes here, we're looking for a GFP positive cell that has a ERG negative nuclei and is BRDU positive for a proliferating cell. And what we see in comparison to controls to mutants at both 11.5 and 13.5 is there's really no difference in the percent of pericyte nuclei that show BRDU staining. So no difference in proliferation of pericytes between controls and mutants at the two different time points. We then thought, well, maybe it's because the endothelial cells are hyperproliferative. And to do this, we did an experiment where we obtained from Science Cell, we obtained human brain microvascular endothelial cells as well as human brain pericytes. And in the pericytes, we knocked down ALK5, and then we waited 72 hours and then collected the conditioned medium from these cells as well as to cells that were treated with scrambled siRNA oligos. And then we took this conditioned medium and cultured it with endothelial cells for 48 or 72 hours. And we saw that there was around a 2.5-fold increase in proliferation of the endothelial cells that were cultured with this conditioned media from the silenced ALK5 pericytes at 48 hours, as well as an increase at 72 hours. Here we did a similar assay, except for instead of assaying proliferation, we assayed migration, and we saw the endothelial cells that were cultured with conditioned media from silenced pericytes migrated around 2-fold more. And this was done with a Bowden chamber, and then here we did it with in matrix gel Matrigel, and we can see that there was an increase in invasion in endothelial cells that were treated with conditioned medium from the silenced parasites. So something about the silenced parasites is inducing the endothelial cells to be more proliferative and migratory. And so we then looked in vivo and see if this was true as well. And so the next two slides will basically show that the endothelial cells in the germinal matrix of the mutants are hyperproliferative. So at embryonic day 11.5 in the germinal matrix, we stained sections with IB4, which marks endothelial cells, PI, which is a nuclear marker, and Ki67, which is a marker of mitotic cells. And you can see that there was an increase in the mutants in terms of their Ki67 marked nuclei in both at embryonic day 11.5 and 13.5, and this is quantified over here on the right. And then we did it again, and with again controls and mutants. This time, right before 6 hours before euthanizing the mice, we injected BRDU, and here we're again staining with BRDU, isoleucin B4, and ERG. And now we want to look at ERG positive nuclei because we're interested in endothelial cells. And we can see that there was a robust upregulation at both embryonic day 11.5 and 13.5 of the percent of endothelial nuclei that are BRDU positive, as quantified here, around a twofold increase. And so this suggests that there's increased proliferation of the endothelial cells. And perhaps this explains why there's decreased coverage at this time point. Now, these findings were specific to the germinal matrix. In vessels in the cortex, when we looked in mutants compared to controls, you can see here that there was no change in the coverage of the endothelial cell vessels by the parasites, no change in the vessel area, lumen area, or vessel diameter. And in terms of proliferation of the endothelial cells, by again staining with BRDU, ERG, and IB4, we saw no difference in the proliferation of the endothelial cells in the cortex. So this difference is specific to the germinal matrix. And then we wanted to query the basement membrane, which is an important part of the blood-brain barrier. And so we looked at collagen 4, which is an integral part of the basement membrane. And in these mice that had the CRE, had the MTMG reporter, and were either flox over plus or flox over flox for ALK5, we looked at collagen 4 staining, the basement membrane marker. And we saw that in mutants compared to controls, there was a marked reduction in staining for the collagen 4 paravascularly. Here we're showing the parasites in green. And so the coverage, if you will, of the collagen 4 of the parasites was markedly reduced in the mutants. But this finding again was specific to the germinal matrix compared to the cortex. In the cortex, we didn't see a reduction in collagen 4 levels. And so this may help explain why the hemorrhage is limited to the germinal matrix. We then isolated cells from the embryonic brain at embryonic day 13.5 from mutants as well as to controls. And so we isolated endothelial cells as well as isolated mural cells. And we looked at the MMPs, matrix metalloproteases. And we were interested in this because we thought that these may be playing a role in degrading the components of the extracellular matrix. And so in the endothelial cells, we saw an upregulation in mutants compared to controls of MMP2 and MMP14. And MMP9 was up, but I wasn't reached statistical significance. In the mural cells, we saw MMP3 was up, MMP9 and 13 and 14. And so we then chose to focus on the enzymes here that have gelatinolytic activity, MMP2 and MMP9. And we looked at their expression by immunohistochemistry. And so again, in the germinal matrix here of controls compared to mutants, we saw that there was an upregulation in the perivascular region around the vessels of MMP9 and MMP2. So that indicated increased levels of MMP2 and MMP9, but doesn't really show anything about their activity. And so what we did next is we did an in situ zymography experiment that looks at perivascular gelatinolytic activities. So these two MMP29 have gelatinolytic activity. They can degrade gelatin. So we took control sections, germinal matrix sections from controls and mutants, and onto these sections we added the substrate DQ gelatin. And so this substrate, when the gelatin is metabolized, will uncage a fluorescein fluorophore, which will then fluoresce. And we see that in the mutant here, but the control, we don't see that because there's not a high gelatinolytic activity as there is in the mutant. And this was, again, specific to the germinal matrix. We didn't see this in the cortex. Now, this gelatinolytic activity that's upregulated in mutants, when we treat with inhibitors of the broad inhibitor of matrix metalloproteases, BB94, betimistat, this could attenuate the gelatinolytic activity, as well as the more specific recombinant TIMP3 could block this. So here we'll focus first on betimistat, and then we'll get into TIMP3. So what we did then is we then moved towards studies in vivo, where we took mice that were either of the control or mutant phenotype, again, the Cree with the ALK5 flocks over flocks, and treated them with daily IP injections of BB94, starting at E10.5. And we analyzed these mice at 12.5, and we saw the mutants that were treated with BB94 had a reduction in their brain hemorrhage. Or if we analyze at E13.5, again, what Julie saw was a reduction in the brain hemorrhage in these mutants. And then if we looked in sections of the brains of these mice at embryonic day 13.5, you can see the mutants without an inhibitor have germinal matrix hemorrhage, as we showed previously, but with treatment with BB94, there's a marked reduction in the hemorrhage. And if we look in cross sections, in sections by staining with CDH5 and NG2, these are coronal sections, what we see is in the mutants, there's, without inhibitor, as we showed before, there was a reduction in pericyte coverage, but this is attenuated. There's improved coverage with treatment with BB94, as you can see here, and is quantified here, the pericyte coverage of the endothelial vessels. So what about TIMP3, the more specific inhibitor of metalloproteases? And so what we started with here is we took human brain parasites and we knocked down ALK5, and this is where the arrow is here shows the ALK5 specific protein band on a Western, and when it was knocked down, that band went away, as well as did phospho-SMAD levels were markedly decreased. And so that reduces ALK5 levels at the transcript level as well, as we saw previously, but also now we're showing that TIMP3 levels are markedly reduced. And then if we do the complementary experiment where we treat with TGF-beta1 in these human brain parasites, we see that TIMP3 levels increase as we treat for increasing amounts of time. And so this was all done with human brain parasites. And then we isolated parasites from mice, either the control mice or the mice that are mutants that have ALK5 deleted in parasites. And we saw that ALK5 deletion in parasites, the isolated parasites had reduced ALK5 as expected, and they also had reduced TIMP3. So all this suggests that TGF-beta is regulating TIMP3 through the ALK5 receptor. And then we looked at TIMP3 by in situ hybridization with an antisense probe, and we could see that there was TIMP3 expressed in the areas of interest in the germinal matrix at embryonic day 11.5. And so finally, as we come to the end here, we took the mutant embryos here that were PDGF receptor beta Cree ALK5 flox embryos. We wanted to see what was the effect of treating with recombinant TIMP3. So we took pregnant dams that were generally ALK5 flox over flox and mated them with PDGF receptor beta Cree ALK5 flox over plus males and treated the dams with daily injections of recombinant TIMP3 or with vehicle, which was PBS, between embryonic day 10.5, 11.5, and 12.5, and analyzed the embryos a day later at 13.5. And what you can appreciate is there was a marked reduction in the brain hemorrhage and those treated with the recombinant TIMP3, the hemorrhage area was markedly reduced. And on sections, you could see a marked reduction of the intraventricular hemorrhage, as well as the hemorrhage in the germinal matrix. And then when we looked by staining on coronal sections, you could see that, of course, those mutants that were treated with vehicle had a reduced coverage of the endothelial cells by pericytes, but when we treated with the recombinant TIMP3, this improved the coverage of the vessels. And we looked at the various vessel parameters, the vessel area, the lumen area, the vessel diameter. All of these abnormalities in the mutants was attenuated by treating with recombinant TIMP3. And then finally, which seems to be a critical part of the phenotype, is the increased endothelial cell proliferation that we see in mutants. When we treated the mice with the recombinant TIMP3 for the 3 days before analyzing at embryonic day 13.5, we saw that this increased proliferation in the endothelial cells was substantially reduced when the mice were treated with TIMP3. And so that kind of ends the data that I wanted to show you. So in summary, what we've shown is that signaling through the ligand TGF-beta interacts with the heterodimeric receptors ALK5 and TGF-beta receptor 2 on Pericyte here. This leads to the phosphorylation of SMAD2/3. They recruit SMAD4, go into the nucleus, and induce expression of TIMP3. The TIMP3 is then secreted, and MMP2 and MMP9, in the absence of TIMP3, meaning when ALK5 is deficient, or when TIMP3 is less than normal levels, we'll get a degradation of the basement membrane, which leads to excess endothelial cell proliferation, a hypermigratory phenotype, and we think these findings are critical in the breakdown of the blood-brain barrier. And finally, the neonatal hemorrhage, which is really a devastating disease in human neonates. So finally, I'd like to just call out a special thanks to Julie here. She really did a ton of work for this, not only the original submission, but the revision, and she was acknowledged by Developmental Cell. And so if you want to meet her, so to speak, you can go to the website, the URL that I'm listing here. And so finally, thanks to Julie, as well as Taya, who initiated this work, Scott, who was a very helpful collaborator in terms of the in situ hybridization experiments, helped to train the other members of my lab that are listed here, the Yale Cardiovascular Research Center and Departments of Medicine and Genetics at Yale, which is a great place to work, the mice, the investigators that supplied transgenic mice, and our funding. And so we'll have a question and answer session now, but if there are any other follow-up questions, feel free to email me and perhaps we can talk as well. Thanks very much.
Thank you, Dr. Greif. I think we have a couple of questions from the audience, so I'm going to turn this over to Dr. David, Joy David, the author, who we're privileged to have today, and she can direct these questions to Dr. Greif.
Hi, Dan. That's a great talk. And I have a couple of questions from the audience here. The first one is an interesting question, which goes as, in several slides in the study, ALK5 flocks-over plus mice were used as a control as opposed to a wild-type mice. So why did we use ALK5 flocks-over plus mice, and do they have any phenotype compared to the wild-type?
Right, so that's a good question. Yeah, we looked at that in the beginning, and we didn't see any phenotype in terms of the brain hemorrhage and some of the vessel parameter that we looked at in terms of comparing wild-type mice versus flocks-over plus with the Cree. We used it mostly because of the ease of breeding. It's easier to just compare litter mates that are flocks-over plus to flocks-over flocks.
Okay, I hope that answered the question. And I have one more question, actually one more after this. Others have described a seemingly similar defect when members of Wnt pathway or GPR124 are deleted. And in some cases, these perturbations in TGF-beta pathway were also described in these mutants. So what the observation was that there were some vascular tufts which had elevated SMA expression. So in our phenotype, do we see such tufted vessels? And have we looked at regulation of Wnt pathway and GPR124 in our mutants?
Yeah, I think we have looked at some of that, and it looks like there's a similar kind of histological phenotype in terms of the tufts that we're seeing with our mutants. We haven't specifically in these studies looked at Wnt signaling, but we think it may be a common phenotype.
Okay. Then I have one more question. The pericyte proliferation, as we showed, is unaffected in the mutants. And do you think the pericyte reduction is a consequence of increased apoptosis or reduced recruitment? And as for the endothelial cells, since the weak adherent staining appears to be preserved, is there any other, is there a morphology of the endothelium affected?
I see. So we didn't see a change that we're clear about in the number of pericytes. We actually didn't, as far as I know. Julie may know better than I do, and you can chime in, Julie. But I don't think we actually looked at the number of pericytes. We just looked at proliferation,
Right.
And we didn't see a change in proliferation.
Right.
We did see. Go ahead, Julie.
We didn't look at apoptosis either.
Right, and we didn't look at apoptosis. Another question was morphology of endothelial cells. I mean, I don't think we rigorously looked at that. I mean, again, Julie, do you want to chime in? What do you think?
Yeah, we haven't looked at it really closely, but what we found was that the endothelial junctions were very elongated and very spindly. That was the only thing we noticed. Other than that, they were actually, yeah, they were preserved, but we showed that they are leaky. So that's one thing. So we didn't do electron microscopy to see how leaky these were. But yeah, as far as the morphology, we just know that the junctions are leaky and they are extremely long and stringy and dilated.
Yeah, I would just add one other thing. I think in the paper that in the paper that I alluded to in during the talk. I'm getting a lot of feedback. Feedback. Julie, do you want to mute yourself now?
Sure.
Okay, maybe that's better. Yeah. So in the paper that I alluded to in the talk, it's a Nature paper from 2010, I think from Christer Betscholt's lab, where they show actually, I'm not sure if this is Christer Betscholt's or Ben Barris's lab, but I think it's Ben Barris's lab actually. They show increased transcytosis with in the deletion of pericytes, the increased transcytosis in the endothelial cells. So if I remember, they don't have a big endothelial cell phenotype per se, but they just show increased permeability by basis of transcytosis. So, okay. Any other questions?
Yes, there are a couple more actually. I'll be really quick. So one question is that did we see a phenotype in any other areas of the body except the GM vessels and other organs which are abundant in PDGFR beta expression? Why is the phenotype preferentially in the brain?
Yeah, that's a good question.
Yeah,
so we did look. Again, Julie, I think you should mute. So we looked in these regions here in the heart, lung, and gut, and we did a number of H&E stains, as well as some other areas, and we didn't see hemorrhage in the other areas. So there's something about the germinal matrix that's specifically prone to hemorrhage, and I think that's why, you know, it's a complication in these prenatal infants. And so there's a number of thoughts about why that is. They have reduced pericytes that we talked about. They maybe have reduced the TGF-beta1 ligand. Their extracellular matrix is thought to be fragile. They have less fibronectin. They have dynamic and high flow, blood flow in that area. So those are some of the reasons why it's thought that that's a particular area that's prone to hemorrhage. But that's a very insightful question and really a critical question that needs to be looked at more closely.
I have
Yep.
two more questions.
Yep.
One is, how come the increased number of endothelial cells that we see in the mutants are associated with vascular leakage?
Yeah. Don't think that we really know per se why increased numbers of endothelial cells are associated with leakage. And I mean, we show that they're hypermigratory, and there's a issue with their junctions. So maybe you know, they lose their junctions become impaired, and then that is really where the leakiness comes from. But the linking proliferation and the leakiness or hemorrhage is not 100% clear. But perhaps there's some kind of inhibition to proliferation when you have these junctions intact, and maybe that gets released when the junctions are relieved. Now, this is complete conjecture on my part. I don't know data that supports that, but that seems logical. Next question.
Okay, and I have one last question, and that is, how did we isolate the murine pericytes, and how was the purity?
Yeah, so that we looked at quite closely, as I remarked in the beginning, that we had to make sure that we weren't marking other cell types and how to isolate them. So we generally use the PDGF receptor beta Cre and a Cre reporter, either MTMG or ZS, and then we isolated the fluorescent marker, and then we also negatively selected, this is by fax, and then we negatively selected against an endothelial cell marker like CD31 or vehic adherent. So we were making sure that we weren't pulling down endothelial cells along with the parasites. Okay.
Great, that's all from my side.
Okay, thanks very much, everybody.
Thank you, Julie and Dan. We still have a few minutes. If there's any other questions, if members of the audience want to raise their hands, then I can call their name. While they think about it, I do have a question for Dan. And it's the TGF signal, especially through ALK receptors, has been involved in HHT, in hereditary hemorrhagic telangiectasia. And I know your approach has been embryonic, but do you have any insight on, you know, what would happen if you delete ALK after birth? Do you expect to see similar abnormalities or abnormalities linked to HHT in some level?
Yeah, I think it depends on what cell type you're deleting it in. If you do it in smooth muscle cells, I mean, then you get actually an aneurysm phenotype, aortic aneurysm phenotype. I don't know, you know, if you did it in endothelial cells, I guess you'd have to do it postnatally in endothelial cells, because if you do it embryonically, you're going to get in endothelial cells, you're going to get a you know, they're going to die early. So yeah, I don't think that's been done. I think, as far as I know, we looked a little bit. Julie's done a little bit of experiments, and again, she can chime in if she wants. And looking in the brain postnatally with the Pericyte Cre-ER to delete in parasites in the brain, and that seems to maybe have a very mild phenotype compared to the in the brain compared to this deficiency during development.
So do you think TIMP3 may be a good therapy for perhaps endothelial cells or other cells that have mutant ALK signaling?
Yeah, I mean, I think that's a possibility. So there's a paper that I think was in Science Translational Medicine a little bit before our paper was published that looked in traumatic brain injury, and showing that I think that they showed us giving a recombinant TIMP3 maybe attenuate some of the brain injury for that. So it'd be something to think about. I mean, you know, these TIMPs are not that specific. So I imagine you'd be affecting a lot of pathways if you tried it during development. So we probably have to refine the pathways involved to try to get to some more specific targets that we could go after.
All right, thank you so much. So
Thank
we
you.
do not have any other questions from the audience. So I hope you found this webinar as interesting and beneficial as I did. These webinars are brought to you by the NAVBO Education Committee. Our next webinar is July 11th. We're going to have Vicky Bouch speak from the University of North Carolina, Chapel Hill. So look for the details on the registration information in the NAVBO Newsbeat. And please fill out the evaluation form at the end of the webinar or when you receive it in a subsequent email, and let us know what you thought of today's webinar and any topics you may want to see in a future NAVBO webinar. Thank you, everyone, and have a good day.
Thank you.